Bismillahirrahmanirrahim. Assalamu alaikum. Here is Mubin Naz, and I'm from MK Preparations. Today we're going to discuss about animal-like protists, the protozoans. All the topics included in this Kingdom Animalia and the protists, the topics included in this have been taken from different source books like FSC Biology Part 1 and Part 2, Federal Biology Part 1 and Part 2, Zoology MILLAR and HARLEY book, Campbell Biology book. And all of these topics are very much important for the preparation of the competitive exams like Punjab Public Service Commission, PPSC, Federal Public Service Commission, FPSC, Sindh Public Service Commission, SPSC, Khyber Pakhtunkhwa Public Service Commission, KPPSC. So the contents we're going to discuss about includes evolutionary perspective of protozoans, life within single plasma membrane, protozoan taxonomy and classification, phylum Sarcomastigophora, phylum Labyrinthomorpha, APICOMPLEXA, microspores, phylum Acetospora, phylum Myxozoa, and phylum Ciliophora. So one by one, we're going to discuss the details of all of these 7 phylums that are under the kingdom Protozoa. First of all, evolutionary perspective of the protozoans. So the fossil record indicates that virtually all the protists and the animal phyla living today were present during the Cambrian period about 550 million years ago. Ancient members of the archaea, that are the bacteria, were the first living organisms on this planet, and the archaea gave rise to the Kingdom Protista. The Kingdom Protista is also known as the Kingdom Protoctista, and they arose about 1.5 billion years ago. Most scientists agree that the protists probably arose from more than one ancestral archaean group. That's why they are grouped under polyphyletic. So zoologists recognize between 7 and 45 phylums of the protists. Groups of the organisms believed to have had separate origins and are said to be polyphyletic, having different origins with different ancestral history or lineage. Some protists are plant-like because they are primarily autotrophic. They can produce their own food by the process of photosynthesis. But others are animal-like because they are heterotrophic and they cannot synthesize their own food. They are dependent on the food from the other organisms. Life within a single plasma membrane. So protozoans display unicellular or cytoplasmic organizations. Often they are more complex than any particular cell in higher organism. Most of these organisms are unicellular, but their unicellularity show some kind of complexity in the structures and the functions as well. So some protozoans phyla, individual groups form colonies. Colonies are the associations of the individuals that are not dependent on one another for most of their functions. So mostly the protozoans exist in unicellular organization form or in the form of colonies. But to be in the form of colonies really doesn't mean that their cells are dependent on each other. Within one colony, every cell is independent on the other. Protozoan colonies can become complex, with more individuals becoming so specialized that differentiating between a colony and a multicellular organism becomes difficult. So within the colonies, the complexity causes the individuals not to distinguish them from the multicellular organisms. They work in a so arranged manner that distinguishing between the colonies and the multicellular organisms becomes really different. Maintaining homeostasis. So we'll have a diagram of one of the protozoan, and one by one, we're going to discuss about their structures and the functions. First of all, A regular arrangement of the microtubules called the pellicle underlies the plasma membrane of many protozoans. As you can see here in the diagram, this one is a protozoan, and just beneath the plasma membrane, a particular arrangement of the microtubules is present that is known as the pellicle. Now, the pellicle is rigid enough to maintain the shape of the protozoan, but it is also flexible. So this one is the function of the pellicle, that pellicle maintains the shape of the protozoans, but it is also flexible. Now, the cytoplasm of the protozoans is differentiated into two regions, as we have an example of the amoeba here, where I'm going to differentiate the cytoplasm into regions. The first region is the ectoplasm. The portion of the cytoplasm just beneath the pellicle is called the ectoplasm, and it is relatively clear and firm. As you can see here in the diagram, just beneath the pellicle, and this one is the region, the ectoplasm. Entirely, this one is the cytoplasm, and it is differentiated into ectoplasm and endoplasm. Ecto means outside, that is present outside and just beneath the pellicle, and this cytoplasm is very clear and firm. Now, the inner cytoplasm is called the endoplasm, and it is more granular and more fluidy in nature. As you can see here in the diagram, this one is the central endoplasm, and it's having a lot of granules, so it's more granular. The conversion of the cytoplasm between these two states, the ecto and the endoplasm, is very much important in the protozoans locomotion, because the conversion of the ectoplasm and endoplasm leads to the formation of the pseudopodia. As you can see here, these are the extensions. These are known as the false feet of the protozoans, so that they can move. Now, most marine protozoans have solute concentrations similar to that of their environments, means they are maintaining the higher solute concentrations both into and outside of the environment. Freshwater protozoans must regulate the water and the solute concentrations of their cytoplasm. So the freshwater protozoans need to regulate their solute concentrations. Water enters the freshwater protozoans by the osmosis because of the higher solute concentrations in the protozoan than in the environment. So in case of freshwater protozoans, their bodies are having higher solute concentrations, but in the external environment, there is higher water potential. So from higher to the lower area, water moves by the process of osmosis. Now, contractile vacuoles remove the excess water. So for the freshwater protozoans, in the body of the freshwater protozoans, excess water is going to be accumulated, and they have a mechanism to remove or to get rid of this excess water by the contractile vacuoles. So they are having the contractile vacuoles, as you can see in the diagram. This contractile vacuole is involved in the removal of the excess water, and this structure is present in freshwater protozoans only. This one is very much important point related to your exam. In some protozoans, contractile vacuoles are formed by the coalescence of the smaller vacuoles. So the contractile vacuoles are mostly formed by the assemblage of the smaller vacuoles. Most protozoans absorb dissolved nutrients by the active transport or ingest the whole food through the endocytosis. So these are their modes of the nutrition. Mostly by the process of active transport across their body, they absorb the nutrients, and in some cases, they ingest the whole food through the process of the endocytosis. Some protozoans ingest food in a specialized region that is analogous to mouth, and this one is called as cytopharynx. Just like in humans, the mouth is present. So in protozoans, the mouth is being replaced by the cytopharynx. This one is the cytopharynx. It just works as the human mouth. It's going to ingest the food particles and to make the food vacuoles. Now the food vacuoles fuse with the enzyme containing lysosomes, and then the lysosomal enzymes are going to break down the food particle, distributing the products of the digestion throughout the body. After digestion is complete, the vacuoles are called as egestion vacuoles, and these egestion vacuoles are going to be released out from the body through the cytopage. So they release their contents by exocytosis, sometimes at a specialized region of the plasma membrane or the pellicle that is called as cytopage. So the cytopharynx is a structure to ingest the food particle, and the cytopage is a structure to release the digestive contents by the process of exocytosis. These 2 points are very much important for your exam, the cytopharynx and the cytopage. Because protozoans are small, they have a large surface area in proportion to their volume. So this high surface area to volume ratio facilitates the 2 other maintenance functions like the gaseous exchange and the excretion. In protozoans, gaseous exchange involves acquiring oxygen for cellular respiration and eliminating the carbon dioxide produced as a byproduct. Excretion is the elimination of the nitrogenous byproducts of protein metabolism, and in case of protozoans, the excretory product is ammonia. Both gas exchange and the excretion occur by diffusion across the plasma membrane through the general body surface of the protozoans. Now, the asexual reproduction in protozoans is by the binary fission. So, both asexual and the sexual reproduction occur among the protozoans. Asexual reproduction is by binary fission. Binary means 2 and fission means breakdown. So in this case, one body of the protozoan is going to split into 2 bodies. In binary fission, mitosis produces 2 nuclei that are distributed into 2 similar sized individuals when the cytoplasm divides. As you can see here in the diagram, the nucleus is going to be divided into 2 parts, and initially the nucleus is going to be divide, and then the cytoplasm will divide, making the 2 individuals, the 2 daughter cells. During the cytokinesis, some organelles duplicate to ensure that each new protozoan has the needed organelles to continue life. Now, depending on the group of the protozoans, cytokinesis may be longitudinal, as you can see here in the diagram, leading to the formation of the longitudinal individuals, or it might be transverse, as you can see here in the diagram. Asexual reproduction by budding. So it's a second form of asexual reproduction in protozoans. Now, what is budding? During budding, the mitosis is followed by the incorporation of one nucleus into a cytoplasmic mass that is much smaller than the parent cell. So you can see here the incorporation of nucleus into a cytoplasmic mass, and then this structure is known as the bud. This bud is going to separate and develop into a new individual. Bud separates and develop into the new individual. Now, here we have a third type of asexual reproduction, that is multiple fission or schizogony. So, multiple fission or schizogony occurs when a large number of daughter cells form from the division of a single protozoan. Schizogony begins with the multiple mitotic divisions in a mature individual. So you can see here, in a mature individual, multiple mitotic divisions will lead to the formation of a lot of nuclei. When a certain number of the nuclei have been produced, the cytoplasmic division results in the separation of each nucleus into a new cell, as you can see here in the diagram. Then each nucleus is going to be incorporated into a separate cytoplasmic mass, forming the new individuals. So this one is the multiple fission, means the splitting of one body into a lot of daughter cells. Now we have sexual reproduction of protozoans. Sexual reproduction requires the gamete formation and then leading to the fusion of the gametes to form the zygote. Now, in most protozoans, the sexually mature individual is actually haploid, and the gametes are produced by the process of mitosis, and then meiosis follows the union of the gametes. So it means the individuals are, the sexually mature individuals are haploid. And if they are haploid, so for sure, their gametes are going to be produced by the process of mitosis. And after the union of the gametes, the meiosis, after the union of the gametes, the next step is the meiosis, so that the new individuals that are being produced, they might be haploid. Now, the symbiotic lifestyles. More protozoans have symbiotic lifestyles, and symbiosis is an intimate association between the two organisms. Now we have different forms of symbiosis. The interaction involves a form of symbiosis called the parasitism. And this one is the first form of symbiotic lifestyle, the parasitism, in which one organism lives in or on a second organism that is called the host. The host is harmed usually and sometimes survives at least long enough for the parasite to complete one or more life cycles. So parasitism is a relationship between the host and a parasite. In this case, the parasite usually gets advantage, but the host is usually harmed. Some parasites have life histories that involved multiple hosts, means they involve more than one host. Parasites live in more than one host, so they complete their life cycle, different stages of their life cycle in different hosts. So the first one is the definitive host. Definitive host harbors the sexual stages of the parasite, and the second one is the intermediate host. The sexual stages may produce the offsprings that enter another host called an intermediate host, and in the intermediate host, they reproduce asexually. So this one is very much important point. The definitive host harbors the sexual stages of the parasite, and the intermediate host harbors the asexual stages of the parasite. These 2 points are very much important regarding to your exam. Now, for the symbiotic lifestyle, we have some other associations. So the commensalism, it's a symbiotic relationship in which one member of the relationship benefits and the second member is neither benefited nor harmed. Mutualism, it's a symbiotic relationship in which both partners get benefit. Now we have protozoan taxonomy. So the protozoologists, these are the scientists or the zoologists that are going to study the protozoans. Now regard the protozoa as a subkingdom, and it consists of 7 separate phyla, which are the phylum Sarcomastigophora, phylum Labyrinthomorpha, phylum Apicomplexa, phylum Microspora, phylum Acetospora, phylum Mixazoa, and phylum Ciliophora. Now we're moving towards the details of these phylums separately. So the classification of the subkingdom into the phyla is based on the types of the nuclei, mode of the reproduction, mechanism of the locomotion, and overall the members of the protozoan species exceed 38,000. These are the 3 bases on which the subkingdom is divided into 7 phylums. So we have classification according to the Miller and Harley 5th edition, according to which you can see here these are the phylum Sarcomastigophora. This phylum Sarcomastigophora is divided into the 2 subphylums, subphylum Mastigophora and the subphylum Sarcodina. The Mastigophora includes Phyto and ZOOMASTIGOPHORA, and the phylum Sarcodina includes the superclass RHIZOPODA and the superclass Actinopoda. Now we have some other subphylum Opalinita. This one is the 3rd subphylum of the phylum Sarcomastigophora. Now these are the remaining 7 phylums of the protozoans: Labyrinthomorpha, Apicomplexa, Microspora, Acetospora, Mixozoa, and Ciliophora. Now we have classification according to the Miller and Harley 10th edition. So according to this, these are the supergroups, the supergroup excavata, and the supergroup excavata includes the suspension feeding individuals, and their first rank and their examples have been mentioned here. Fornicata, Parabacellia, Euglenozoa, and these are their respective examples. Similarly, the second group is the Amoebozoa, which includes the amoeba, and their first ranks are Tubulini, Acanthomoebidae, and Entomoebidae, along with their examples. Now, the third group is the Rhizopoda, which includes the pseudopodia containing individuals, the foraminiferans, and the radiolarians. The chroma alveolata, it includes the cryptophyci, heptophyta, staminophiles, and alveolata, along with their respective examples. So this one is the difference in the classification of protozoans in Miller and Harley 5th and 10th edition. So it would be helpful for you to study the examples along with their groups in both the 5th and the 10th editions of the Miller and Harley. So you can cope up with your questions in the exam. Now, first of all, the phylum Sarcomastigophora, it includes 18,000 species. Phylum Sarcomastigophora containing 18,000 species. This one is the largest protozoan phylum, having the following characteristics. The first one is that they are unicellular or colonial. They show locomotion by the flagella, pseudopodia, or by both structures. They have single type of nucleus. They show sexual reproduction usually. They might be autotrophic, and they might be saprozoic, living in decaying organic matter, or they might be heterotrophic. They obtain energy from the organic compounds. So these are the general characteristics of the phylum Sarcomastigophora with 18,000 described species. Phylum Sarcomastigophora is divided into 3 subphylums: subphylum Mastigophora, Sarcodina, or Pylinata. One by one, we're going to study the details of these subphylums. Subphylum Mastigophora, they show flagellar locomotion. Subphylum Mastigophora, showing the flagellar locomotion, is divided into 2 classes, class PHYTOMASTIGOPHORA and the ZOOMASTIGOPHORA. Now we're going to study the details of these classes. Initially, class PHYTOMASTIGOPHORA possess the chlorophyll and 1 or 2 flagella. And examples include the Dinoflagellates, Euglena, Volvox, and Chlamydomonas. Now, we have marine Phytomastigophorans. The example of the marine Phytomastigophorans includes the Dinoflagellates. Di means 2. They are having 2 flagella. One flagellum wraps around the organism to cause to spin on its axis, and the second flagellum is a trailing flagellum that pushes the organism forward. In addition to the chlorophyll, they are having some other pigments that are known as the xanthophyll pigments, which give them golden brown color. The Red Sea is named after the toxic dinoflagellate blooms, and the several genera, such as the Gymnodinium, produce toxins. Periodic blooms of these organisms are called the red tides and result in fish kills along the continental shelves. Now we have freshwater Phytomastigophora, which includes Euglena. Each chloroplast in Euglena has a pyrenoid which synthesizes and stores polysaccharides. So in the diagram, you can see here the pyrenoid structure which synthesizes and stores polysaccharides. If cultured in the dark, euglenoids feed by the absorption and lose their green color. Usually, Euglena have ability to photosynthesize, but if they are going to placed in the dark without any light, so they become heterotrophic. Some euglenoids lack chloroplast and are always heterotrophic. Euglena orient towards the light of certain intensities. And why they orient towards the light of certain intensities? Because they have a pigment shield stigma that covers a photoreceptor at the base of the flagellum. As you can see here in the diagram, they have pigment shield stigma that is going to cover the photoreceptor at the base of the flagellum. Euglenoid flagellates are haploid and reproduce asexually by the longitudinal binary fission. Sexual reproduction is unknown in case of Euglena. Now, the freshwater Phytomastigophora volvox. Volvox is a colonial flagellate. Colonial flagellate consisting of up to 50,000 cells embedded in a spherical gelatinous matrix. So there are up to 50,000 cells in one colony of the volvox. Individual cells possess two flagella, which cause the colony to roll and turn gracefully. Now we're moving towards the volvox reproduction. Reproduces asexually as well as sexually. So first of all, asexual reproduction occurs in spring and summer. When the parental colony dies, it ruptures and releases the daughter cells. So this one is the mechanism of asexual reproduction. Sexual reproduction in volvox occurs during the autumn. Now, some species are dioecious, they're having separate sexes, and others are monoecious, having both sexes within one colony. In autumn, the specialized cells are going to differentiate into micro and the macro gametes. Macrogametes are large, filled with the nutrient reserves and non-motile. As we can see in the diagram, the macrogametes are large, filled with the nutrient reserves and are non-motile. Okay. Microgametes form as a packet of the flagellated cells that leave the parental colony and swims to a colony containing the microgametes. Okay, so macrogametes and the microgametes. Macrogametes are large, filled with the nutrient reserves. The microgametes are small, okay, and they are formed as a packet of the flagellated cells. Now the microgametes leave the colony and they will move towards the macrogametes. The packet then breaks apart and the syngamy occurs. As you can see in the diagram, the packets break apart and syngamy occurs between the macro and the microgametes. Then the zygote, an overwintering stage, secretes a resistant wall around itself and is released when the parental colony dies. Because the parental colony consists of the haploid cells, now the zygote is undergoing meiosis so that the next individuals in the colony might be haploid. Now, class ZOOMASTIGOPHORA, which includes TRYPANOSOMA, TRICHONYMPHA, TRICHOMONAS, Giardia, and Leishmania. Class ZOOMASTIGOPHORA lack chloroplast and they are heterotrophic. If you remember, initially we studied about the class PHYTOMASTIGOPHORA. So PHYTOMASTIGOPHORA, they were autotrophic. They were capable of doing photosynthesis. But the class ZOOMASTIGOPHORA, they are not capable of doing photosynthesis. These are heterotrophic. They are important parasites of human. We have one species, Trypanosoma brucei, that is divided into 3 subspecies. The first one is TB brucei. This one is the parasite of the non-human mammals of Africa. TB gambians, it causes sleeping sickness in humans. TB rhodesians, it causes sleeping sickness in humans. CC fly, these are the intermediate hosts and the vectors for all of these 3 subspecies of the Trypanosoma brucei complex. Now we have life cycle of the Trypanosoma. Trypanosoma multiply asexually in the gut of the fly. The fly is CC fly. So in the gut of the fly, they are going to reproduce asexually for about 10 days and then they migrate to the salivary glands. As you can see here, they're going to multiply asexually in the gut of the fly for about 10 days and then they're going to migrate towards the salivary glands. While in the fly, trypanosomes transform in 15 to 35 days to the mature individuals. When the infected CC fly bites an other vertebrate host, the parasite travel with the salivary secretions into the blood of the new definitive host. As you can see here in the diagram, these are the trypanosomas in the blood vessels of the new definitive host. The parasites multiply asexually in the new host and again transform through a number of the body forms. And these parasites may infect the blood, lymph, spleen, central nervous system, and the cerebrospinal fluid. When trypanosoma enter the central nervous system, they cause general apathy, mental dullness, and lack of the coordination of the individuals. Sleepiness develops and the infected individual may fall asleep during normal daytime activities. So the trypanosoma usually cause sleeping sickness in humans. Now, subphylum Sarcomastigophora. This one is the second subphylum of phylum Mastigophora, Sarcomastigophora. So these include the pseudopodia and the amoeboid locomotion. Subphylum Sarcomastigophora, the pseudopodia and the amoeboid locomotion. When feeding and moving, they form temporary cell extensions that are called the pseudopodia, as you can see here in the diagram. Pseudopodia exist in a variety of forms: the lobopodia, philopodia, reticulopodia, and exopodia. Lobopodia. Lobopodia are broad cell processes containing the ectoplasm and the endoplasm and are used for locomotion and engulfing food, as you can see here in the diagram. Philopodia. Philopodia contain ectoplasm only and provide a constant two-way streaming that delivers food in a conveyor belt fashion. So these are philopodia. Reticulopodia. Reticulopodia are similar to philopodia except that they branch and rejoin to form a net-like series of the cell extensions. So these are the reticulopodia. Exopodia. Exopodia are thin, filamentous, and supported by a central axis of microtubules. So these are exopodia. The cytoplasm covering the central axis is adhesive and movable. Food caught on the exopodia can be delivered to the central cytoplasm and amoeba, as you can see in the diagram. Subphylum Sarcomastigophora is further divided into the classes superclass Rhizopoda, to which the class Lobosea having genus Amoeba belongs. The second is the superclass Actinopoda, to which the foraminiferans, heliozoans, and radiolarians belong. So first of all, superclass Rhizopoda, containing 4,000 species. The species includes the amoeba, entamoeba, nigellaria, archela, Diphylgia, foraminiferans. So superclass Rhizopoda, the class Lobosea, the naked amoeba. Naked, they are having no tests or shells, so they are normally found on the shallow water substrates of the freshwater. Other members of the superclass Rhizopoda possess a test or shell. Tests are the protective structures that our cytoplasm secretes, and test might be calcareous, made up of calcium carbonate, proteinaceous, made up of protein, siliceous, made up of silica. So the amoeba are of two types. They might be naked without any test or shell, or they might be shelled or test organisms having shells made up of the calcium carbonate or the protein or the silica, or the chitinous made up of chitin. Now, the superclass Rhizopoda and the class Lobosea. Now we're going to discuss about the shelled amoeba. So first one is the archela. It's a common freshwater shelled amoeba. It has a brown proteinaceous test that is flattened on one side and domed on the other side, and the pseudopodia project from an opening on the flattened side. Number two is Diphylgia, that is another common freshwater shelled amoeba. Its test is vase-shaped and is composed of mineral particles embedded in a secreted matrix, as you can see here in the diagram. Now, the Entamoeba histolytica. It causes one form of dysentery in humans, and the symptoms of the dysentery are the diarrhea. It causes inflammation and ulceration of the lower intestinal tract. Now we have superclass Actinopoda with 3,000 species, which includes the heliozoans. So it includes the foraminiferans, heliozoans, and radiolarians. So first of all, foraminiferans. These are marine group of amoeba. This one is a diagram of foraminiferans. They possess reticulopodia and secrete a test made up of the calcium carbonate. Number two, heliozoans. These are aquatic and either planktonic or live attached by a stalk to some substrates, as you can see. Haliozoans are either naked or enclosed within a test. Radiolarians, these are planktonic marine and freshwater amoeba. So they possess a test made up of the silica of long movable spines and needles of the ornamental lattice, as you can see in the diagram. Now, the phylum Labyrinthomorpha. So they are spindle-shaped, non-amoeboid vegetative cells showing gliding motion, and most members are marine. Some are saprozoic or parasitic on the algae or seagrasses. Phylum Apicomplexa with 5,500 species, including gregarines, monocystis, Coccidians, Eimeria, Isospora, Sarcocystis, Toxoplasma, or Plasmodium. So phylum Apicomplexa, they are all parasites, and their characteristics include they have apical complex for penetrating the host cell. They have single type of nucleus. They have no cilia and flagella. And their life cycles include the asexual stages and the sexual stages. The asexual ones include the schizogony or the sporogony, and sexual stages include the gametogony. So class Sporozoi, including the Plasmodium and the Coccidians, life cycle has the three phases: schizogony, gametogony, sporogony. Now, what is schizogony? These stages are very much important for your exam. Schizogony is the multiple fission of an asexual stage in the host cell to produce many smaller individuals that are known as the merozoites. The schizogony to produce these merozoites is called merogony. Now, the second stage is gametogony. So the merozoites undergo gametogony, and the parasites forms either microgametocytes or the macrogametocytes. The microgametocytes undergo the multiple fission to produce biflagellated microgametes, and macrogametocytes develop directly into a single macrogamete without any multiple fission. Now, a microgamete fertilizes a macrogamete to produce a zygote, and zygote now becomes enclosed in an oocyst. Now, the sporogony. The zygote undergoes meiosis and resulting cells divide repeatedly by mitosis. This process is called the sporogony. Sporozoites infect the cells of a new host after the new host ingests and digests the oocyst or sporozoites and otherwise introduced by a mosquito bite. Now we have species description of Plasmodium and Coccidians. So initially Plasmodium. It causes malaria. The symptoms of the malaria are called paroxysms. Chills and the fevers are the symptoms of the malaria, and it leads to the rupture of the red blood cells and release of the toxic metabolites. Now, the life cycle of the Plasmodium. It involves the vertebrate and the mosquito hosts. Now, schizogony occurs first in the liver cells and later in the red blood cells, and gametogony also occurs in the red blood cells. So initially, when an infected mosquito is going to bite a host, a vertebrate host, so it injects sporozoites. This point is very much important. The infected mosquito, when it's going to bite a vertebrate host, it will inject the sporozoites. Okay. Now the sporozoites infect the liver, enter in the red blood cells, and in the red blood cells, the schizogony takes place. Schizogony is the multiple fission of these sporozoites to produce the merozoites. Now inside the red blood cells, the gametogony takes place, and it leads to the formation of the gametocytes. A mosquito takes in gametocytes during a meal of the blood, and the gametocytes subsequently fuse. Okay. Now these gametocytes are engulfed by the mosquito when it's going to bite a vertebrate host. The zygote. Now the zygote penetrates the gut of the mosquito and transforms into an oocyst. Okay, so this one is the gametogony. Now we're moving towards the sporogony. So in the sporogony, the haploid sporozoites are going to be formed that are going to enter in the new host by the bite of the infected mosquito. So these are the three stages. The schizogony, the formation of the merozoites from the sporozoites. The gametogony, formation of gametocytes from the merozoites. And then the sporogony, formation of sporozoites from the gametes or the zygote or the oocyst. Number 2, coccidiosis. So coccidiosis is primarily a disease of the poultry, sheep, cattle, and rabbits. They have two genera, Isospora and Eimeria. These are the parasites of poultry especially. Cryptosporidium has well known with the advent of the AIDS. It causes chronic diarrhea in AIDS patient. So it is only a known protozoan to resist the chlorination and is the most virulent in the immunosuppressed individuals like the patients of the AIDS. Congenital lexoplasmosis is a major cause of the stillbirths and the spontaneous abortions. Now we have phylum Microspora with 850 species, and they are obligate intracellular parasites. They parasitize beneficial insects. The example includes Dinosema bombicus. It parasitizes the silkworms, causing the disease pebrine. Okay, and the Nosema apis, it causes dysentery in honeybees. Now, phylum Acetospora, it includes the Paramixa and Haplosporidium. They are also obligate extracellular parasites. Haplosporidium are parasitic in the cells, tissues, and the body cavities of the mollusk. Now, the phylum Myxozoa have 1250 species. They are obligate extracellular parasites in freshwater and marine fish. They have a resistant spore with 1 to 6 coiled polar filaments. Myxosporion is a Myxosoma cerebralis, which infects the nervous system and auditory organs of the trout and salmon, causing the whirling or the tumbling disease in the trout and the salmon. Now the phylum Ciliophora with 9,000 species includes the Paramecium, Stentor, Euplotes, Vorticella, Blantidium. Okay, so phylum Ciliophora includes the Paramecium, as you can see in the diagram. The characteristics of the Ciliophora include cilia for the locomotion and for the generation of the feeding currents. On the entire surface of the Paramecium, you can see here fine hairs that are known as cilia. Number 2, they are having relatively rigid pellicle and more or less fixed shape. Okay, so they are having rigid pellicle just beneath the membrane. Number 3, distinct cytosome mouth structure is present. Okay, so distinct cytosome mouth structure is present. Dimorphic, they're having dimorphic nuclei. They're having nuclei of two types, the micro and the macro nuclei. The larger one is the macro and the smaller one is the micro. Macro is one and the micro are many. Cilia and other peculiar structures of the Paramecium or the phylum Ciliophora. Cilia are small, similar to flagella, except that they are much shorter, more numerous, and widely distributed on the surface of the protozoans. Cilia join to form cirri. Okay. Trichocysts are peculiar structures primarily used for the protection. As you can see in the diagram, they are rod-like or oval organelles oriented perpendicular to the plasma membrane. Now the nutrition of the Ciliophora. So Paramecium have a ciliated oral groove along the one side of the body. Cilia of the oral groove sweep small food particles towards the cytopharynx. Some free-living ciliates prey upon other protists or small animals. Suctorians are the ciliates that live attached to their substrates. Now the genetic control of the Paramecium. Ciliates have two kinds of nuclei. A large polyploid macronucleus regulates the daily metabolic activities, as you can see here in the diagram, and one or more smaller micronuclei. These are the genetic reserves of the cell. Now the reproduction. Ciliates reproduce asexually by the transverse binary fission and by the budding. And the diagrams you have already discussed initially in case of Euglena. Ciliates reproduce sexually by the process of conjugation. Okay, so the partners involved are called the conjugates. Sticky secretions of the pellicle facilitates the adhesion of the two partners that are the conjugates, and the ciliate plasma membranes then fuse and remain fused for several hours. The macronucleus does not participate in the genetic exchange. Instead, the macronucleus breaks up during or after the micronuclear events and reforms from the micronuclei at the end of the division in the daughter ciliates. After the separation, the X-conjugates undergo a series of the nuclear divisions to restore the nuclear characteristics of the particular species, including the formation of a macronucleus from one or more micronuclei. Now, the cytoplasmic divisions that form daughter cells accompany these events. Now you have a diagram here, as you can see. These are the conjugates, okay? The process of conjugation has just started here. So this one is the macronucleus and that one is the micronuclei. So the macronuclear degeneration and the macronuclear degeneration and meiosis of the micronuclei will take place here. Now, in this case, the mitotic division of the remaining pronuclei will take place, leading to the micronuclear migration and their fertilization. And after the fertilization, they are known as the X-conjugates. And the diploid zygote nucleus is now undergoing the micronuclear multiplication. And now, the beginning of the nuclear modification leads to the cell division and the nuclear segregation, ultimately forming the 4 daughter cells from the 2 parent cells. Okay, so we have done with all the topics of the protozoans, all the phylums of the protozoans. Now we have some important questions. So the first one is, they are broad cell processes containing the ectoplasm and the endoplasm and are used for the locomotion and engulfing food. So these are Lobopodia. Number 2, which is very small phylum that consists of protozoa with spindle-shaped non-amoeboid vegetative cells. So these are Labyrinthomorpha. Number 3, Entamoeba histolytica causes dysentery, that is known as the amoebic dysentery in humans. Number 4, it is a common freshwater shelled amoeba. It has a brown proteinaceous test that is flattened on one side and domed on the other side. So that one is Archela. Number 5, which one parasitizes silkworm? So this one is the Nosema bombicus. Number 6, which specialized region in the protozoa is analogous to mouth? So that one is the cytopharynx. Number 7, which of the following do not belongs to the class PHYTOMASTIGOPHORA? So this one is Myxosoma cerebralis, because the volvox, Dinoflagellates, and the Euglena, all of these are phytomastigophorians. Number 8, which of the following is not a type of the pseudopodia found in the amoeba? So this one is the Lophopodia. That one is not a type of the pseudopodia. Number 9, which of these groups is characterized by the cells that have more than one nucleus? So these are ciliates. They are having 2 types of nuclei, micro and the macronuclei. Number 10, the agents of the red tide, which actually may be red, brown, yellow, or even colorless. These are dinoflagellates. Number 11, excretory product in the protozoans is ammonia. Number 12, flagella is present in Mastigophora. Number 13, which of the following group have shell? So these are RHIZOPODA. Number 14, egestion in protozoans takes place by cytopyge. Number 15, sporozoites of the Plasmodium are formed in oocyst. Now we have some past paper questions from the PPSC zoology or biology exams. The first one is, average number of the cells in volvox colony are 50,000. And this question has been taken from the PPSC zoology paper 2017. Number 2, the continuing and the intimate association between the 2 different species is called symbiosis. And this question has been taken from the PPSC zoology paper 2020. Number 3, contractile vacuole in protozoa is basically concerned with osmoregulation. The contractile vacuole is used to get rid of the excess water in the freshwater protozoans. And this question has been taken from the PPSC Zoology exam 2003, 2006, as well as 2009. So this question is very, very, very much important and might be expected to come in your next exams. Number 4, it is exclusively a parasitic group. So the sporozoa, these are exclusively parasitic in nature. This question has been taken from the PPSC Zoology exam 2004. Number 5, fossils of which of these first organisms were found? So these are protozoans. And this question is also taken from 2004. Number 6, cytopharynx is the structure present in the paramecium. Okay, so this question has been taken from the PPSC Zoology exam 2005. Number 7, Entamoeba histolytica is a member of the class none of these. So it doesn't belong to any of the given classes mentioned here. So this question has been taken from the PPSC Zoology exam 2006. So we have done with the entire topic. We, inshallah, will come up with your next topic. Until then, Allah Hafiz.